Hello everyone, welcome to the ITLS Academy. Today's topic is Introduction to Enzymology. So let's start our topic. First of all, we have to know what is enzyme. See the amazing facts about the enzyme. Did you know that enzymes are what? Enzymes are nothing. Enzymes are just a natural tiny catalyst and they are capable of accelerating the chemical reaction by millions of time. Enzymes is simply a natural tiny catalyst. What they are? They are tiny catalyst, and these tiny catalysts are natural. Okay, these tiny catalysts are naturals and they are used to accelerate the chemical reaction by millions of time. If you see the definition of the enzymes, so enzymes are the biological molecules. They are what? They are biological molecules and they catalyze the biochemical reaction without involvement. Without involvement. Okay, this is the definition of the enzyme. Suppose in any reaction there is an enzyme A whose weight is 2 gram is involved in that reaction, but when a product is formed, this A enzyme is released and the weight of these enzyme will be 2 gram because it does not involve in the reaction. Now see the importance of the enzyme. They play an essential role in metabolism, signaling and various cellular process. As we all know that enzymes are play a vital role in a metabolism reaction and they give a signaling. Signaling means that they provide a signal to the catalyst that they work or not, or what is the rate of the working of the enzyme, what is the rate of the working of the catalyst. And it also play a vital role in a cellular process, means that when more than one cell are involved in any reaction and these involve in any reaction and these reactions are on the cellular level, like for example cellular communication. In cellular communication, more than one cells are involved. Now see the historical milestone of the enzymes. Pepsin is the enzyme. It is developed in 1836 by Theodore Shevon. To the in 1913, Maud Menton and Leonard Michael evolved the enzyme kinetics. Enzymology has a rich history of scientific inquiry and discovery, but these two discoveries are very important and basic discoveries. Now, what is the structure of the enzymes and what is the function of the enzymes? As we all know that enzymes are made up of proteins. What enzymes are made up of? Proteins. Okay, and these proteins are form 3D complex and play a vital role Now this is the basic of structure of enzymes and function of the enzyme. Now we are going to see in details. See what is the amazing fact about the structure and function of the enzymes. Enzymes are a molecular machines. Enzymes are the molecular machines which interact three-dimensional structure and determine the specific function. This is the amazing fact about the structure and function of the enzyme. See, enzymes, what is the structure of the enzymes? Enzymes are typically consist of protein chains. Okay, that clear that enzymes are made up of proteins and they are folded like that. They form a complex shape and these shapes are 3D shape, three-dimensional structure, including active sites where a substrate binds. Suppose this is your enzyme structure, okay, and this is your enzyme and this is your active site, okay. So active site is also present in the enzyme whereas any substrates are bind, okay. This is the structure of the enzyme. Now what is the function of the enzyme? Enzyme lower the activation energy required for a chemical reaction. As we all know that when any chemical reaction happen, then it's required lots of energy. And what is the work of the enzyme? Enzyme lower these activation energy and accelerate the reaction rate. Like suppose A plus B is form AB without involvement of enzyme, it take 2 hours. Now we introduce enzyme C. C is what? C is any enzyme. Now what happened? This time decreases and become 1 hour. Okay. It is simply an example. And it also lower the activation energy. Now see the specificity. Each enzyme exhibit markable specificity, recognized and binding to a specific substrate molecules. Each enzymes are very specific. Suppose enzyme A have to bind with substrate B, then its active site are very specific that it can only bind with substrate B. This is the enzyme specificity. Enzyme cofactors and coenzymes. Enzymes always require some cofactors and coenzymes. They are additional things involved with the enzyme to bind with the substrate or increase the chemical reaction. Now see what is the amazing fact behind the coenzymes and cofactors. Enzymes often require helper molecules. Okay, enzymes can't work alone. They always require what? They require helper molecules, and these molecules are cofactors and coenzymes. When these helper molecules join with the enzymes, they play a function optimally. What is cofactor? Cofactors are inorganic ions. See the difference between the cofactor and the coenzymes. Where coenzymes are the inorganic ions and cofactors are the inorganic ions and coenzymes is organic molecules. Okay. Or cofactors is also a small molecule that assist enzyme activities by participating in catalyst. Cofactor assist the enzyme activities. They are not bind with the enzyme, but they do what they do. They assist the activities of the enzyme by participating in the catalysis. Okay. For example, metal ions like Mg2 plus, magnesium ion. This is what? This is magnesium ion. Okay. But where coenzyme are organic compounds and they bind to the enzyme. Cofactor only assist the activities of the enzyme, but coenzyme bind with the enzyme and then participate in the catalysis. How they participate in the catalysis? By donating or accepting chemical groups. Like example NAD plus and coenzyme Q. Now next is enzyme kinetics. See, enzyme kinetics are totally depend upon a Michaelis-Menten equation. See, enzyme kinetics are totally depend upon the Michaelis-Menten equation. Clear? What is this equation? This is equation said that V is equal to V max in bracket S Km plus bracket S multiply by V. See, this is the Michael-Menten equation. V is equal to V max in bracket substrate Km plus in bracket substrate and V, where V, triple V, like this V and this V is the initial reaction velocity. Okay, V is what? V is initial reaction velocity. V max is equal to maximum velocity, and in bracket S is a substrate concentration, and Km is nothing, is just a Michaelis constant. Okay, here V max is the maximum velocity, S is the concentration of the substrate, Km is the Michaelis constant, and this V is a initial reaction of velocity. This is about the Michaelis-Menten equation. The equation also describes the relation between the substrate concentration and the reaction velocity. What is the relation between the substrate concentration and reaction velocity is described by the Michaelis-Menten reaction. It helping us understand enzyme substrate interaction. It also helps us to understand that how the enzyme and a substrate are interact with each other. Now, enzyme substrate complex. How enzyme bind with any substrate and what type of complex are formed. Suppose I already told you that suppose this is your enzyme. Okay. And this is your substrate. Now, this is your substrate and this is your enzyme, and this side is the active site of the enzyme. Here lock and key method are applied. These enzyme go and bind with the substrate. Okay, enzyme and when these enzyme bind with the substrate, here a enzyme substrate complex is formed. Enzyme substrate complex is formed, and these enzymes of substrate complex then converted into a product, and along with the product enzyme are released. Okay, and now see the introduction part. Enzyme substrate complex formation is a crucial step in each enzyme catalyzed reaction. When an enzyme is used to catalyze the reaction, this is the most crucial part when the enzyme bind with the substrate and form an enzyme substrate complex. Where the enzyme bind to its substrate to facilitate the conversion of reaction into a product. When these enzyme bind to the substrate, then form a enzyme substrate complex, and at that point the enzyme react with substrate and form a product, facilitate to conversion of the reactant into a product. Amazing fact behind the enzyme substrate complex is that enzyme can enhance reaction rate by strategizing factor of millions, making them essential catalysts in a biological process. This is the amazing fact behind the enzyme substrate complex. Now see how these enzyme substrate complex is formed. It divided into 3 steps. First step is substrate recognition, in which the active site of the enzyme recognize the binding site of the substrate molecule, and after that they form a enzyme substrate complex. First of all, the active site of the enzyme have to recognize that where the binding site of the substrate molecules are present, because because for the binding of enzyme to the substrate, it is important that binding site of the substance should be recognized. Then the enzyme substrate complex are formed. Amazing fact, enzyme active sites are highly specific, allowing them to bind to a particular substrate with a precision. They are highly specific and they only allow to a particular substrate to bind with it. They are not bind with every substrate, but their active sites are very specific and they are only allow only a specific type of substrate to bind with it. Now, when these substrates are recognized by the active site of the enzyme, then the formation of the complex take place. Once bond, the enzyme substrate complex undergo a conformation changes, aligning reactive group of the catalytic reaction take place. Now, what is the amazing fact behind this? Enzyme often undergo induced fit upon substrate binding, adjusting their structure to accommodate the substrate. Active site of the enzyme also adjust their structure so that it can easily bind with the substrate. Now, third part is catalysis. The enzyme catalyze the conversion of the substrate into a product. Now, the role of the enzyme is started where the enzyme catalyze the reaction. They allow the substrate to convert into a product and it also facilitate the rate of the reaction by lowering the activation energy barrier. When the activation energy become lower, the rate of reaction will be increases. Now, what is the amazing fact behind it? Enzyme do not alter the equilibrium of a reaction but significantly accelerate the attainment of the equilibrium. Suppose it is A plus B is equal to AB and the amount of A will be 2 gram and amount of B will be a 3 gram. When we added enzyme 1 gram, but it not affect the equilibrium. Here, if the reactant site is 5 gram, then the product site will also be 5 gram. It manage the equilibrium between the reactant and product side. Only the work of the enzyme is that it accelerate the reaction. Okay. Now this is all about the how enzyme substrate complex are formed. What is the step behind it? What is the mechanism? All about the enzyme substrate complex formation. Now next is significance. The formation of the enzyme substrate complex allow enzyme to perform their catalytic function with remarkable specificity and efficiency. When enzyme and substrate complex are formed, then it allows the enzyme to perform their catalytic function, and these catalytic function will be remarkable specificity and efficiency. Understanding this process is fundamental to elucidating enzyme kinetic and designing enzyme-based therapies and biotechnologies application. And then they are work in a enzyme-based therapies and biotechnology applications. Now see which factors are affecting the enzyme activities. First factor is your pH, temperature, substance concentration. These all the factors which affect the activities of the enzyme. See what is the amazing fact behind the enzyme factor affecting the enzyme activities. Enzyme activities can be modulated by various factors like pH, temperature and substrate concentration. If we see the first point, that is pH, we all know that enzymes have an optimal pH range for their activities. Every enzymes have optimal pH range for their activities. If any deviation in that pH, then the activities maybe decreases and maybe these enzymes become denaturation. Okay, so every enzyme requires optimal pH range for their activities. Same with the temperature. Every enzyme exhibit optimal activities at specific temperature. If the temperature vary, then these enzymes become denaturation. Now third is your substrate concentration. Enzyme activity typically increase with substrate concentration until reaching saturation. When the saturation reaches, the enzyme activities also stop, but before the saturation, enzyme activity is directly proportional to the substrate concentration. If we increase the substrate concentration, the enzyme activity will also be increases. This is about the factor affecting the enzyme activity. Now, enzyme inhibition. Word inhibition means that the compound, the material which inhibit the enzyme activities. Amazing fact behind the enzyme inhibitor is enzyme activity can be regulated by inhibitors. Molecule that bind to an enzyme and alter their activity. There are enzymes, there are inhibitors which bind with the enzymes. Suppose this is your enzyme and this is your substrate. Now these substrate have to bind to the enzyme and convert it into a product, but sometimes there are some inhibitors which bind to the enzyme other than a substrate, and when these inhibitors bind to the enzymes, it alter the activities of the enzyme. It alter the activities of the enzyme. Okay. Now there are two types of inhibitor. One is competitive inhibition and other is non-competitive. In competitive inhibitors are those inhibitors that compete with the substrate, and after competition they bind to the active site, and they are often resembling the substrate structure. They are competitive inhibitors are resemble with the substrate. That's why they are bind to the active side of the active side of the enzyme. Now some are non-competitive inhibition. The inhibitor that bind to the allosteric side. In enzyme there are, suppose this, there are allosteric sites are also present, as the non-competitive inhibitors bind in the allosteric site rather than a active site, and when these bind with the allosteric sites, they alter the activity of the enzymes. Now case study. Discovery of a penicillin, a competitive inhibitor of bacterial enzyme involved in cell wall synthesis. This is the example of the competitive inhibitor. Now difference between the competitive inhibitor and non-competitive inhibitor is only like that. Competitive inhibitors are those inhibitors which bind in the place of substrate and it binds with active site of the enzyme. And non-competitive inhibitors are those which bind in the allosteric site of the enzyme and alter the activity of the enzymes. Now enzymes regulation. See, amazing facts behind the enzyme regulation, that is, cell titry regulate the enzyme activity to maintain the homeostasis and response to changing the environmental condition. Allosteric regulation and covalent modification. Binding of a regulatory molecule to a allosteric site on enzyme altering their activities. That I already told you that the molecules bind with the allosteric site of the enzyme and alter their activities. Now second is your covalent modification. Covalent modification means that addition or removal of chemical group that affect the enzyme activities. Suppose there are some group that may be added or they may be removed from the reaction, and these addition and removal of the chemical group alter the enzyme activities. Like case study, the role of insulin in regulating glucose metabolism through allosteric activation of enzyme involved in glycolysis. And what is the role of enzyme in biotechnology? Amazing fact behind the role of the enzymes in biotechnology is that enzymes are indispensable tools in biotechnology during advancement in medicines, agriculture and industry, which means that now enzymes play a very vital role in a biotechnology in which advancement in medicines, agriculture and industry. What is the application of enzymes in industrial enzymes are used in food processing industries, detergent industries, biofuel production and pharmaceutical synthesis. Biomedical application of the enzyme, that enzymes play a vital role in diagnosis, test, therapeutic drugs and gene editing technologies like CRISPR case 9. Okay. Now what is the roles of enzymes in medicines? See, enzymes have revolutionized medical treatment, from enzyme replacement therapies to a target cancer therapy. They play a vital role in a medical field also, from enzyme replacement therapies in which the enzymes of a body is replaced by the other enzymes, and targeted therapy in a cancer treatment in which enzymes are used to target the cancer cell. Enzyme replacement therapy, treating enzyme deficiency by administering purified enzymes. Suppose you have a deficiency of a insulin. So what happens? You become a diabetic. So for the treatment of the diabetes, what should we do? We administrate the purified enzyme in the place of deficient enzyme and treat the diabetes. Now what is targeted therapy? Targeted therapy inhibiting the specific enzyme involved in disease pathway like protease inhibitors for HIV. This is about the role of enzyme in medicines. Emerging trends in enzymology. See what is the emerging trend in enzymology. Enzymology is a dynamic field with ongoing research driving innovation in enzyme discovery and engineering and application. Now, enzymology is a very dynamic field in which the research are ongoing, the innovations are ongoing, the enzyme discovery is also ongoing. Directed evolution, engineering enzyme with desired properties through an iterative round of mutation and selection. And metabolic engineering are those engineering which modify the enzyme pathway in organism to produce valuable compound or enhance the metabolic efficiency. Synthetic biology design novel enzymes or enzyme system for specific applications such as biosensor or bioremediation. In synthetic biology, what should we do? We design a biosensor or bioremediation. In biosensor or bioremediation, we design a novel enzymes or enzyme system which play a specific role in biosensor and bioremediation. Enzyme is sustainable agriculture. What is the role of enzyme in sustainable agriculture? Enzyme offer promising solution for sustainable agriculture. What is the solution behind the sustainable agriculture? Like they improve the soil health. What they means? Enzyme are used to improve the soil health. It also improve the nutrient uptake and crop yield. Overview, soil enzymes. What are soil enzymes? Enzyme produced by soil microbes. There are lots of enzymes which are produced by soil molecules which play a crucial role in nutrient cycle, organic matter decomposition and soil fertility. Enzyme supplement. Soil enzymes are different and enzyme supplement are different. They are applying enzyme supplement to the soil or crop can enhance the nutrient availability. Soil enzyme produced by soil microorganism, but enzyme supplements are used as a supplement to the soil crop which enhance the nutrient availability, reduce the fertilizer requirement and mitigate the environmental impact. This is about the enzyme. What is the role of enzyme in sustainable agriculture? So this is a time of cues. Which of the following is not a factor that can affect the enzyme activities? Please give your answer in the comment box. First question is, which of the following is not a factor that affect the enzyme activities? A, pH, temperature, substrate concentration, molecular weight. Second question is, what is the primary function of enzyme? Energy production, catalyzing chemical reaction, maintaining cell structure and DNA replication. Now third question is, what does the Michael constant Km represent in a Michael-Menten equation? A, it represents the maximum reaction rate. B, it indicates the enzyme specificity. C, it measures the substrate concentration at half maximum velocity. D, it determines the pH optimum for a enzyme activities. So give your answer in the comment box. And if it is all about the enzyme, introduction to enzymology. If you have any query, please let me in a comment box, or you can follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn, and you can also download our app from a Google Play. Thank you so much. If you have any query, this is a helpline number. Thank you.